Okay. So hopefully we are kind of settling into our new normal of being at home doing this learning. And the things that we did so far in the last couple weeks were things that we were going to do exactly the same, more or less, anyway. So, um, the next concept, however, is a little bit more challenging, I think. It's going to involve some math. So it'll be a little different. So I want to kind of create like this kind of flipped lecture sort of thing to do, and then I'm going to be doing some problems with you. And again, that's a little bit more difficult to kind of do in this sort of environment. So I'll probably do some practice problems. Some of you might get this after a couple problems. Some of us might need 10 problems to go through. Some of us might still have questions, and that's okay. So I'll probably do like a Google Hangouts for you to answer questions on that shortly after this is posted. So I'm going to probably give you a lot in this lesson, and hopefully you might not necessarily need everything, but it's there if you want to go back and do it. And for this lesson here, if you could print it, that might be good. Or if you want to, you don't necessarily have to print it, but you can follow along on paper and do your calculations on paper. That might be easier. Or maybe try to do like a split screen. That might be a little bit more challenging, but we'll just kind of have to make do with what we have. And I think that if you spend some time with this, you'll be able to get it. So we're going to be learning about here is something called microevolution. And this is a little bit different than what we have been talking about before, which is macroevolution. And macroevolution is looking at like big changes that can be observed between species rather than little changes that can be observed within a species. So what microevolution is looking at the changes. And I'm just getting used to using this stylus again, this little tablet. So you're going to have to bear with me. Sometimes it's a little laggy. So is the changes in gene frequencies that happen over time. Let me add something to this. Changes in gene frequencies within a population. All right. So our big question here is, how do we know if a population is evolving? And can we look at some data and see if a population is evolving? Is it evolving quickly? Is it evolving slowly? That sort of thing. So what I usually start this unit on is looking at a question from the Ayers test. And, I would probably have you do this one in kind of on your own. It's a drag and drop sort of problem. Since we don't know what's going on with the Ayers test, it's still a good problem to go through kind of as an introduction, but I'm not going to necessarily assign it as like an eduelastic or anything like that right now. More to come on when we find out what's going on with testing. So, but I still, like I said, I still like this problem, and I want to go through it as kind of an introduction to this concept of gene frequencies. So what it says here is we have a butterfly, and that butterfly has two phenotypes. It has both white individuals and yellow individuals, saying that the white allele is dominant. So I'm just going to kind of do some kind of note taking down here, and I'm going to use W. Oh no, they use A, so I could use W, but let me just stick with what they do. So they say that we have capital A represents the white allele. So if an individual is white, it can either be homozygous for white or it could be heterozygous. We don't know just looking at that butterfly what its genotype is. So that's what we're going to try to look at and figure out how you would look at populations, be able to determine that information. But we do know that either the homozygous dominant or heterozygous genotypes are going to be white phenotypes because the white is dominant. And the yellow is recessive. So little a is going to be yellow, okay? So that's what they're telling us here. And for however they got it, I'm not sure how they got this, but they have that you have, these are the gene frequencies of the population to start with. That there is 30% of the individuals are homozygous for their white wings, the white butterfly, and 50% of them are heterozygous. So overall, the population is 80% white and 20% yellow. Okay. Oops, I was meant to just highlight that, not to show that, but I guess I could just highlight this one as being the yellow one. The other two are the white phenotype. And I have to stop touching my face. So what it's telling me here is that there's a predator introduced that prefers the yellow trait, meaning that predator likes to eat yellow butterflies. And it wants you to make a prediction based on that information about what's going to happen to these gene frequencies. And the way that this works usually on the ARES test is you do a lot of dragging and dropping. I'm just going to write it up because this is just in my PDF. It's not like a dynamic thing like it would be. Again, I don't want to spend too much time on this particular problem. Just a good intro. Okay, so what we would predict, okay, so it wants you to place the genotypes that will have a higher rate of predation in the blank box. So these are our genotypes here. And it says right here, so this is an easy part. It says they prefer the yellow. So we already did that. Before we even approached a problem, we kind of took some notes on it. So that's a good strategy. And so in this box, it wants us to put the genotype or genotypes, and it's only going to be one genotype. So it's going to be little a is going to be the one that is the genotype that is going to be equal to the phenotype yellow. It doesn't ask me for that there, the yellow part, but that I'm just going to add that. All right, so we have that part done. Usually that'd be a drag and drop. You would just drag that into there. And then it's wants us to make a new population frequency sort of prediction based on that information about the yellow being now more predated. So if the yellow butterflies are now going to be eaten more by a predator, you would predict that the numbers of those would go down, right? And by percentage, then these would have to go up, right? So, but there's a little caveat here when it says, it tells you assume all genotypes are represented. So you might make a prediction that, depending on how much of that predator is there and how much it decides to eat, that this might go down to 0. And that would be, I think, a logical prediction, but it's telling you to assume all genotypes are represented. And you only have these 10 blocks, so you can only do them in like 10% increments. So if this goes down, but it's still represented, the only thing we can do is the minimum amount that you could put there is 10%. So what we have to move is one of these this little a over to here, and I'm just going to draw it in. And that's the only correct answer for the little a, because if you put 2 of them in there, that's 2 out of 10 or 20%. And that means it stays the same. You didn't have a correct prediction there. You want to predict that they go down, but they're still represented. So that's the only correct answer for little a is one of the boxes. Now it doesn't care which box you put it in. So that's fine. Now we have to make a prediction about these two. So if this went down, one of these two has to go up, and or both of them. And so for these two, there's going to be more than one correct answer that could be predicted. And so, you might predict that if these go down, and I don't know, they're just looking at the next generation. So you're not necessarily going to appreciate that eventually there might be less of these because there's less of the little a's. But you could predict that these would go up maybe these would go up. Maybe these would stay the same. So let's see what we have here. We have 9 more boxes we can fill. So I'm going to go ahead and put for big A, as I'm going to put them at, they're definitely going to go up. So I'm going to put them at 40% and see if that makes sense. My little stylus is giving me trouble here. Okay. So they started at 30, and I want to make a prediction that they're going to go up. So I'm going to put them at 40. And then that would mean that I have just the big A, little a staying the same. So I think that's an okay prediction. That is one of the correct answers. Again, there might be more than one correct answer for the dominant genotypes, but you definitely need, the only correct answer for the little a is the way that we have it there, where it is going to be only one of those. Okay, so that's just looking at changes in the gene frequency over time, and just a little bit of time in this case, just looking at a couple different generations. Okay, so the next thing we're going to do is we're going to talk about something called the Hardy-Weinberg principle, and this was looking at changes in gene frequencies and making predictions about if a population was evolving based on if they met this equilibrium. And Hardy and Weinberg independently arrived at this, so that's now called the Hardy-Weinberg principle. They weren't like working together on this. And for ninth grade biology, this is one of the more challenging concepts, I would say, for ninth grade biology. It's some that people struggle with. But if you kind of follow along with me, I think that you'll have an okay time with it. On a quiz or test, I will always give you the equations, and I'm not sure about like an AP test, but we're not in AP right now, so I'm not going to worry about that. So, but you need to know what the different values in the equation represent. That you have to know, and you have to know how to use the equations together when you're approaching a problem. So that's what I'm going to kind of take you through. This kind of little penguin, Professor Penguin, shows you what these different values are, but I'm going to also do it here. So what we're looking at is in a population, and we're just going to look at one trait at a time. So it's a little bit of an oversimplified look at a population genetics. But what the value in the Hardy-Weinberg equations represent is p squared is going to represent your individual's percentage of frequency of your individuals that are going to be homozygous dominant. Okay, so I'm going to use big A there. And then Q squared, and Hardy and Weinberg have like book-long papers going through and how they worked all this out. So it's much more complicated than, I'm not going to get into the derivation of the Hardy-Weinberg principle. We're just going to use the equations to make some predictions about if our population is evolving. So Q squared is going to represent our frequency of our homozygous recessive. And then the way they worked it out is the value 2PQ is going to represent your number of individuals, your frequency of your individuals that are, as you can imagine, P and Q together are different. So that's going to be your individuals that are heterozygous. Okay, and all of those are going to be equal to 1. And also the second equation is the frequency of the dominant allele. I could just use capital A here, and the frequency of the recessive allele are going to be equal to 1. So we're going to use both of these equations to approach problems. And again, kind of follow along with me on a couple problems first, and then I'm going to have you work on your own. This is something called Hardy-Weinberg equilibrium. I'm not sure if I spelled equilibrium correctly, but you get the idea. Equilibrium we've already learned about when we talked more about cell chemistry. The equilibrium is when things are staying the same. So That's what with populations, if things are staying the same, they're not evolving. So there's some assumptions for Hardy-Weinberg where as if you have these things met, your population is not going to be evolving. And in any real population, that's not going to be the case. You're going to always have some of these factors going on. So it's kind of what we call an ideal population versus a real population. We're going to go through these things that are assumptions of the Hardy-Weinberg equilibrium, meaning if you don't have these things present, then your population is likely not going to be evolving. And they're not going to be in any particular order, but and these were all the things that we went through in the previous lecture. So something like, if you have no gene flow, then your population is not going to be evolving. And so that, again, gene flow is individuals going in versus out. Okay. Same thing. If you have no genetic drift. And remember, genetic drift is a chance event, just to review that. That's a chance event that's going to take out members of the population, and it's much more likely to affect a small population. So again, you can never really have no genetic drift, but large populations are going to be less likely to evolve. And I think that kind of makes sense because you can understand that a large population is going to be less affected by chance events. Let's see which one I want to do now. Again, this one never happens, so no populations could be never evolving just based on this one alone. No mutation. So we always have mutation happening. That's kind of like our palette for natural selection. But if you do have mutation, then your population will be evolving, and we always have mutation. But so that one's kind of self-explanatory. And then the next one is natural. Let me change that to no natural selection. Again, not something that will happen in real populations. There's usually going to be something that will be a more favorable trait. So no natural selection. That means that each allele had an equal chance. And we know that's not usually the case. Sorry, this is looking like chicken scratch, but you know my deal. And then the fifth one is no sexual selection. And another word of saying that is that you had random mating. And random mating doesn't usually happen in most animal populations. It happens in plant populations. It happens in starfish. Starfish random mating. Male starfish spawn their sperm. Female starfish spawn their eggs. They meet each other in the ocean. And that's the example of random mating. Whereas usually there's some sort of mate choice that's usually involved in a lot of other animal populations and some plant populations as well. All right, so now I'm going to switch over to some practice problems and go through how to solve a couple of them and then kind of turn you loose on a couple of other problems. So let me pull those problems up here. I think I have them ready to go, so I might not have to stop. Okay, so these are the problems, and I'm going to go through a couple of them, and I think you should follow along with this. You can use separate paper or use your paper, print this, whatever you want to do. And then you can kind of do them on your own and then come back and check them. So the equations will always be given to you by me, but again, you need to know what the values in the equation mean and how to kind of extract the information from the word problem in order to get the correct answers. So in the Hardy-Weinberg, just to kind of review real quick, My computer is being difficult here. Okay, I think we're all caught up here. So in the Hardy-Weinberg problems, remember p squared is going to represent your individuals that are homozygous dominant. Q squared represents your individuals that are homozygous recessive. And 2pq is a value that represents your individuals that are heterozygous. And P represents in the gene pool the number of your alleles, or the frequency of your alleles that are the dominant alleles, and your frequency of individuals that are the recessive alleles. And both of those values are going to be equal to 1. All right, so now I'm going to read this problem here and get some information out. In a certain African population, 4% of the population is born with sickle cell anemia. So 4% is going to be your individuals that are homozygous recessive. So Q, the 0.04 is Q squared. All right, so I'm going to go ahead and write that up here. Q squared is going to be equal to 0.04. And 0.04. You want to have a calculator up when you're doing these problems. That's another important thing to talk about. Just something that could do square roots or or squaring. That's the only two things that you're going to have to do here. Okay, so that'll work for me, I think. All right, so if I want to get Q by itself, I'm going to have to take it's Q squared. So get it by itself, I can take the square root. So if I look here and I put 0.04 and then take the square root of that, I get 0.2. So when Q is going to be equal to the square root of 0.04 or 0.2. So now I have Q, I had Q squared. That's usually what's given to you because that's the only one that you can usually see when you're looking at a population. And Q plus P are going to be equal to 1. So if I have Q at 0.2, I know, of course, that P is going to be equal to what? 0.8. Okay, now I have everything I need in order to go back to this equation. Start here, start at Q squared. This is going to get messy. And then I go to this equation, and then I go back to this one. So, and then I can solve it. So I have P, so 0.8 squared plus 2 times P is 0.8 times Q, which is 0.2, and plus what I already know is 0.04, so I can do Q squared, 0.2 squared, but since I already know that value, when I do these, I usually just put the 0.4 there, but if you put 0.2 squared, you're going to get the same answer. So when you look at this here, this one is actually really easy. You don't even need your calculator, but I'll just do it just to show you guys. And so if I have 0.8 squared, that is going to be 0.64. And then plus, okay, you guys know 8 times 2 is 16, times 2 is 32, 0.32 plus 0.04. And if I did that right, those should all equal 1. So I have 96 and 4. That should equal to 1. So now I have everything, but you want to make sure that you report the way the question's asking, so that we know that individuals in this population, this African population, are 64% of them are going to be homozygous dominant, 32% of them are going to be heterozygous, and 4% of them are going to be homozygous recessive or have the trait. Remember, in sickle cell, there is a heterozygote advantage. So sometimes that happens for genes, that there's a heterozygote advantage. So remember that they will be they'll be more resistant to malaria, but they won't have sickle cell disease. Okay, so that's it. That's all you start with one piece of information, and you can figure out the rest of them. All right, so let's move on to the next problem. And then after we do that problem, you should do these on your own and then come back and check your answers. Okay, so this problem looks like it's not going to be as easy mathematically to solve, but we'll give it a go. There's a wildflower in California called the dwarf lupine, and it normally has blue flowers, but occasionally you'll see pink flowers in the population. And it's controlled at a single gene where pink is completely recessive to blue. In a census of this population, they found that there were only 25 flowers out of 3,316. So this one I definitely need to use my calculator for. And so I have 25 divided by 3,316. So it's giving you some information here, and a lot of times people just start putting these numbers in, but you have to figure out what we have to start with Q squared. So we have to figure out the frequency of those individuals, the percentage of them. 25 divided by 3,316 total flowers. So you need to be careful about what you're doing mathematically here. Oops, what did I do there? Sorry. 25 divided by 3,316 is going to be equal to 0.0075. So I'm going to do some rounding here, and I'm going to say that Q squared, that's what I just figured out, Q squared is going to be equal to, what did I just say? 0.008. Okay, so that's Q squared. So now I got to figure out Q. So what I'm going to do here is I'm just going to take the square root of points. Oh, I should have kept it in there, but I actually would prefer to put my rounded value in there anyways. 0.008, square root it, and I'm going to end up with 0.09. I'm going to round that. So Q is going to be equal to 0.09. So now I have Q, I can figure out P. P is going to be 1 minus 0.09. So that's going to be equal to 0.91. Oops. Oh no. Q, let's rewrite it again. Q is going to be equal to 0.09, and P is 1 minus 0.09 is going to be equal to 0.91. Why is it doing that? I'm not 979. Let me just erase it. I used to do this on the iPad. It was a little bit easier because then I could see what I was doing. This is just like a blind tablet here. 0.91. So remember, these two values are going to be equal to 1. So that makes sense. All right. So I have that information. Now I can figure out what my frequency of homozygous dominant and my frequency of heterozygotes. Now that I have that information about the gene pool. So what I have here is, I want to just make sure I have space here. So P squared, so I'm going to go with 0.91 squared, that's P squared, plus 2 times 0.91 times 0.09 for PQ, plus, I already know this value Q squared to be 0.008. Again, if I put 0.09 squared, I'm going to end up with something that's the same or very, very close because of rounding. All right, so now I have that. So, but I have to figure out the frequency, I have to do the calculation. So 0.91 squared is 0.83. I want to make sure I don't make too many rounding errors. And then I have I have 0.91 times 0.09 times 2 is equal to 0.16. And then this other part here is going to be, we already have that at 0.008. So those are going to be very close to 1. It's not, because of rounding, not quite, but the frequency of your individuals in that population, did they give me a letter, a color to use? They didn't, I don't think, but capital B, 83% of them, if you have that 25 out of that 3,316, it's a small number of individuals that are, so it's like 0.8%. 83 of your population should be homozygous dominant. 16% of your population is heterozygous. And of course, we already know that that 0.8% is going to be homozygous recessive. Okay, so what I want you to do for these next problems is to approach them on your own first, and then to pause the video, approach the problems yourself first, and then come back and check your work. I'm going to go through them just the same way as I did with these, but again, I want you to give them a shot first on your own. Okay, so in this problem, it's telling me that in a university study, 64 of percent, no, not, see, I'm messing up already. 64 out of 400, so definitely not 64%. 64 divided by 400 is 16%. That sounds more accurate there anyways. So I have my value Q squared is, remember, my individuals that are going to be little t, so that is going to be 0.16. So to get Q by itself, I have to take the square root of 0.16, and I'm going to just show you, but hopefully you know the square root of 16. But so 0.16 square root that, and I'm going to end up with my value of Q is going to be equal to 0.4. Now P is going to be equal to 0.6. Remember, those together are going to be equal to the 1. All right, so now I have everything I need to go back to the first equation. Remember, we want to start here, then come down here and look at that one, and then move back to this one, and that's going to be what you want to do. Just need to make sure that when you're doing the math, that you don't stick 400 in here, you don't even stick 64 in there. You have to figure out the frequency of individuals that are homozygous recessive. All right, so now that I have that, I have 0.6 squared plus 2 times 0.6 times 0.4 plus 0.16. Sorry, my little tails that are 0.16. Okay, so again, I don't need my calculator here. You should use it if it makes you more comfortable to check your work, but some of these you don't. The ones I give you on an assessment probably you won't have to, but by all means use it so you can make sure that you're correct. So 0.6 squared is going to be 0.36, and then 0.6 times 0.4 is 0.24, times 2 is going to be 0.48, if that makes sense. And then I have 0.16. So 48, that should be 84, right, together. So we know that that equals 1. So now I can figure out what it's asking me. It wanted to know in this population that they're going to be 48% of them are going to be that are going to be heterozygous. So, but I like to label all of them. So my individuals that are going to be homozygous for their tasting, 36% of individuals, and this is the PTC, classic PTC problem we've talked about. So If you're a taster, you could 36% chance that you are in homozygous dominant for your tasting. 48% of individuals are heterozygous, and then finally we have only 16% of individuals that are homozygous recessive or non-tasters. Okay, so that's how you solve that problem. The next couple, when you approach them, if you haven't already, that one's pretty straightforward. These have a little bit of, these problems are a little bit more challenging, so you might want to give them a shot and then come back, or look at one of them and then go back and do the other one. All right. Again, like I said, not everybody is going to need to see me do like 20 problems in order to get this, but sometimes it's more challenging. And I do agree that this way is going to be not ideal, but it is what it is. Okay, so we have a population of individuals that is 16% Rh negative, which is not too far off from what the national average is, or that. And if you are Rh negative, that's 6.16%. So what they're giving me here, 0.16, this problem should look familiar already because it's going to be very similar to one of the other ones, is going to be q squared. So q squared, again, I always start with q squared. .16. Why is it doing that? This is just as bad as my smartboard. Maybe it's me. It is me. Q squared is going to be equal to 0.16. And then Q is going to be the square root of 0.16. So you know that the square root of 16 is 4. So 0.4. And then 1 minus 0.4 gives me P, which is going to be equal to 0.6. So this is the same problem that we already did, just given to us in a slightly different way. All right, so now that I have Q and P by themselves, I can go back to my first equation. So 0.6 squared plus 2 times, oh my goodness, 6 squared plus times 2 times 0.6 times 0.4 plus, we already know, is 0.16. Again, if I would have put 0.4 squared there, I'm going to end up with the same thing. So 6 squared should be 36 plus 24 times 2.48 plus 0.16. So in a population of 16% of individuals that are Rh negative, 36% of them are going to be homozygous dominant for their Rh factor on their red blood cells. 48% of them are heterozygotes, and again, it was already given to us, 16% of individuals are homozygous recessive or have Rh negative blood types. Okay. Ooh, St. Patrick's Day problem. I must have been to this by St. Patrick's Day one year, so not too far off. I'm recording this on the 21st, so I might have switched some things around a little bit to get this in for the AIRS test when we did it last time. Okay, so this problem is kind of like a real life problem, so I think that we're going to definitely need our calculator more on it. But it's saying that if we assume that 2% of individuals have this recessive phenotype, so Q is going to be equal to 0.02, and then so if I have, just write this off to the side here, Q squared is going to be equal to 0.02. My kids are running amok on the snack box while I am telling them not to bother me while I'm recording. So Yeah, I don't know how your house is going. They're eating me out of all the snacks that we have. Oh. All right. Anyways, that means that Q is going to be equal to 0.002. Take the square root of that is going to give me Q is going to be equal to 0.1. Let me make sure I did that right. 0.02. Oh, see? 0.02. Oh. 0.02 square root. Okay, 0.14. Q is going to be equal to 0.14, and then P would be equal to 0.86. So now I can go here and say that we have 0.86 squared plus 2 times 0.86 times 0.14 plus our 0.02. Okay. And so 0.86 squared is 0.74. So 74% of individuals are going to be homozygous for, again, hair color is not this simple, but if we only had those two phenotypes, brown and red, which we know we don't, but 74% of individuals in that population would be homozygous for their brown hair. 0.86 times 0.14 times 2 equals 0.24 plus that 2%. So 78 or 98 plus 0.02, so those equal to 1. I love when that happens. It all equals to 1, so it checks out. So in this population, 74% of individuals are homozygous for their brown hair, 24% of individuals are heterozygous, and just 2% are redheaded with being recessive. All right, so that's not it for this problem, though. So hopefully you were able to get to at least to that part. And then what it's asking you is that in an Irish population, the incidence of individuals who are heterozygotes is 34%. Now, is that population, is 34% 24%? No, they're not. They're different. So we would say that this population is not in Hardy-Weinberg equilibrium. So there's something interesting going on genetically. Maybe it was, and likely it was a founding effect. So likely it was due to genetic drift or a founding effect when certain individuals, a small set of individuals moved into a certain area. But maybe it was sexual selection. Maybe red hair was a preference or something. But again, we don't necessarily know what that factor is, but we know one of those 5 things is not, that one of those 5 things is happening. Likely more than one of them. Okay, so that's how you approach those problems. All right, and then finally, I think I have this clover problem. Again, looking at actual kind of frequencies when I wrote this problem, I think. So I was actually looking up to see. All right, so assume that 3-leaf clovers are dominant and 4-leaf clovers are due to a recessive gene. And so the incidence of 4-leaf clovers is going to be 1 out of 10,000. So that means that our value Q squared is going to be equal to 0.0001. And so if I want to get Q by itself, I have to take the square root of that, because that's Q squared. Square root of that is going to be equal to 0.01. So that means that P is going to be, of course, equal to 0.99. Okay, now I have everything I need to go back and figure this out. So 0.99 squared plus 2 times 0.99 plus, or times, sorry, 0.01 plus q squared already is 0.0001. And again, I could have put 0.01 squared here. That's fine. I'm going to get the same answer. So I have 0.99 squared is equal to 0.98. And then I have 0.99 times 0.01 times 2 equals 0.02. And that makes sense because this is just with 1 out of 10,000, that frequency is so small. So already these equal to 1, but then that's just kind of a negligible amount here. So when you only have 1 out of 10,000 being a 4-leaf clover, that's a very rare gene in the population. In order to get them both together, it's only 1% of the gene in the population. And the other 99% of the genes in there are going to be the dominant genes. And then, so it's asking you about the population of clovers in my backyard. I did not actually genetically sequence them to find out 8% of them are carriers, but worldwide, 2% of them should be carriers, and these are 8%. So again, maybe something, the original population that seeded my yard with clovers happens to be carried more of that gene, because we would only expect 2% of them to be the heterozygotes, but actually 8, it looks like in this population 8. So all that we can say is that the population is not in equilibrium, not in Hardy-Weinberg equilibrium, because it's not, the frequency is not 2%, it's 8%. Okay, that's it. So I'm gonna probably go on like a Google Hangouts or something to answer any questions that you have for this. So kind of stay tuned for when that's going to be. It'll be, posted, or I would have something with it. So I just don't know, because I'm recording this on the 21st of March. So, next week we have spring break, and then we'll come back, and then I already gave you like a week's worth of work. So stay tuned. I just don't know the schedule yet. Okay, so I hope you guys are doing well. Look forward to hearing from you and maybe seeing you guys soon. Thank you.